Okay, let's get started. Can someone confirm if they can hear me and see my screen as well? Okay, great. Welcome everyone to week three of Introduction to Cell Biology TA course. My name is Yashwant, so we'll basically look at previous year's assignment questions and briefly go over them and how to solve them and everything. And if you guys have any doubts, we can discuss them as well. Okay, so with that, let's jump into it. Let's go through the questions. So the first question reads, which of the following is not explained by Mendel's laws of genetics? Independent assortment, dominance, segregation, and linkage. What do you guys think? Which of the following is not explained by Mendel's laws of genetics? Is it independent assortment of alleles, dominance of alleles, segregation of alleles, or linkage? Yeah, so linkage is what is not explained by the Mendelian laws of genetics. Essentially, what linkage means, as a refresher for those of you who might have not remembered. If you have two alleles that are in very close proximity to each other, what usually happens in a biological setting is those two genes would have a very high probability of uh, getting basically segregated and transferred together. So in this case, if gene 1 and gene 2 are in very close proximity to each other when this chromosome replicates during mitosis or meiosis, gene 1 and gene 2 get, or these are genes in this case, but if you talk about alleles as well, that's the same thing. They get segregated together, so they're not independently assorted. In fact, this is one of the drawbacks of or disadvantages of Mendelian laws of genetics, where it doesn't completely explain the biological phenomenon. Okay, so moving on, a mutant that produces more proteins or proteins that are inactive than wild type is called a what mutant? Is it an amorph mutant, hypomorph mutant, hypermorph mutant, or antimorph mutant? So here they're talking about the different varieties of mutants. So if a mutant produces more proteins or protein more proteins, they can be active or inactive than the wild type. Then that mutant is called what? Is it an amorph protein, hypermorph protein, or antimorph protein? So in this particular case, they're asking you to consider that the protein is active, but it's produced more in number. Any idea? Hypermorph. Okay. Yeah, so essentially it's called a hypermorph mutant. So hyper meaning more and hypo meaning less. So the name itself sort of suggests. So just to briefly go through the different mutants. So if you have don't consider the deletion and duplication part. Let's just look at the homozygote and for the most part wild type uh, part of this figure. So you have a normal wild type that's producing 20 proteins. Amorph nucleus, amorph mutant results in a loss of function. There is no protein that's produced. So if it's heterozygous, only the wild type gene can produce protein then. Overall, less protein. Hypomorph is basically producing less than the normal amount of protein. It's not completely gone, but the amount of protein that's produced by this mutant is reduced. So in this case, so compared to wild type, where 20 proteins are produced by both the alleles, in this case only 13 are produced. And hypermorph is basically producing more than the normal amount. So a mutant that can produce more protein than the normal amount. So 25 proteins are produced in this case. And neomorph is basically a mutant that's producing a completely new type of proteins with a different function. So it's not identical, at least function wise, to the wild type protein. So that's neomorph, and antimorph is again basically a mutant that produces a protein variety that acts against the normal function or impedes the normal function of the protein. Okay, so the next question reads, in Mendelian genetics, if two heterozygous parents are crossed with each other, the phenotypic ratio of the offsprings is what? Is it 3 is to 1, 1 is to 2 is to 1, 1 is to 1, or 4 is to 0? So if two heterozygous parents are crossed with each other, the phenotypic ratio of the offsprings is what? So let's just consider a single gene here, or two different alleles for the gene. Uh, if there are two heterozygous parents for that gene, then what is the ratio, phenotypic ratio of the offsprings? Yeah, so it would be 3 is to 1. Essentially, so in this case, what we have is, let's just consider some allele that codes for the color of a seed. There are two different varieties, white and red, right? So if you cross, so in this case, these are homozygous parents, but even if you have heterozygous parents, if you cross, the phenotypic ratio will always be equal to 3 is to 1. Okay. The idea that different pairs of alleles are passed to offsprings independently is Mendel's principle of unit inheritance, or segregation, or independent assortment, or none of the above. So, in Mendel's laws of genetics, what's the idea of the different pairs of alleles being passed on to offsprings independently of each other called? So, we've briefly discussed this in the first question where we talked about linkage. So, ignoring linkage, what would ideally happen is alleles would be individually passed on to the offspring, independently passed on to the offspring. And what is that called? Is it unit inheritance, segregation, independent assortment, or none of the above? Yeah, now it's called independent assortment, wherein each of the alleles would assort independently and randomly get passed on to the offspring. Yeah, one allele does not influence one allele, or its location does not influence another allele, or its ability to be passed on to the offspring. So that's independent assortment. Okay. The next question reads: a plant with heterozygous alleles has blue flowers, and one of the alleles codes for white flower color. Which of the following statements is true? Blue color is dominant trait, white color is recessive trait, blue color is recessive trait, or both A and B, wherein blue color is dominant and white color is recessive, or true. So, what do you guys think? So, a plant with heterozygous alleles, so it has alleles for both blue color and white color, but the flower color, the phenotype is blue, or has blue flower. Yeah, so even though it has white flower color allele, the phenotypic outcome is blue flower. So then is blue recessive or dominant? Is white recessive or dominant? Yeah, so essentially both A and B are correct, wherein since it has both the alleles for white and blue, but blue is the phenotype, uh, blue seems to be the dominant trait, and white seems to be the recessive trait. So both A and B are correct. Okay. Which of the following statements about enzymes is not true? All enzymes are proteins. Enzymes reduce the activation energy of a reaction. Enzymes participate in cellular metabolic processes. Enzymes speed up a reaction. Which of the following statements about enzymes is not true? Are all enzymes proteins? Do enzymes reduce the activation energy of a reaction? Do enzymes participate in cellular metabolic processes? Or do enzymes speed up a reaction? Which of the four statements is false? Okay, yeah, so all enzymes are proteins is a false statement here. Do you guys know? Okay, can you guys name uh, enzymes that are not proteins? So I've given the structure here. There's only one enzyme that's not a protein. Uh, do you guys know what that enzyme is? Okay, so there is only really one enzyme that's not a protein, and that enzyme is ribozyme. So ribozyme is essentially a complex of either RNA by itself or RNA along with proteins, but the catalytic activity of this complex is always within the RNA. So yeah, ribozyme is essentially the only enzyme that's not a protein. Okay. Which of the following is the most abundant biomolecule on Earth? Is it lipids, proteins, carbohydrates, or nucleic acids? What do you guys think is the most abundant biomolecule found on Earth? So can you please explain the function of ribozyme? Yeah, so essentially ribozymes are they have several functions, but one of the main key function is splicing of RNA and uh, regulation of RNA. So RNA splicing, wherein the introns get chopped away and so on and so forth. So which of the following is the most abundant biomolecule on Earth? Do you guys think it's lipids, proteins, carbohydrates, or nucleic acids? Okay, proteins. Uh, it's not in fact proteins. So even though proteins are vast in number, carbohydrates really outnumber the proteins. So every living thing is made up of carbohydrates as well as most of the non-living things. So like sugar molecules, so on and so forth. Well, I should not say most of the non-living things. Even sugar is non is from living things. So every living thing is made up of carbohydrates. So carbohydrates are the most abundant biomolecule found on Earth. Closely followed by lipids and proteins, but carbohydrates are the most abundant. Okay, so a test cross is performed to determine the genotype of a tall plant. A cross results in equal number of short and tall plants. What is the genotype of the parent plant? So a test cross is performed to determine the genotype of a tall plant. How do you perform a test cross? So you want to know what the genotype of a plant is or some other organism is. And one of the ways you can determine that is by doing a test cross. How do you do a test cross? Does anyone know? How do you perform a test cross? So it's very simple. So all you have to do is, so if you have a plant that you do not know the genotype of and you want to see what the genotype is, all you have to do is cross it with, for a particular function, the recessive the homozygous recessive parent of the plant. So let's say you have some uh, let me see. So let's say you have some plant with allele X. You want to know what X is, whether it's XX or XX or XX, right? So what you will do is you can cross this with plant B, which is recessive for X. So if you cross it with this, so then you'd be performing a test cross, and you can know what type of genetic makeover the unknown plant is. So now they're saying a test cross is performed to determine the genotype of a tall plant. The cross results in equal number of short and tall plants. So then what would be the genotype of the parent plant?
Excuse
Heterozygous.
me, sir.
Yeah.
Sir, till now one assignment answers has not been uploaded in Gmail, sir.
In Gmail as in? I did not understand.
So till now, one assignment solutions have not been uploaded in Gmail to us, sir.
Okay, so this is this year's assignment, the ones that you finished, or previous year's assignment solutions?
So the assignment that we finish now.
Oh, that you finished now. Okay, so you then you'll have to talk to the course organizers. So you can raise a request in the forum and ask them when you can expect the answers. Some courses they give the answers straight away. Some courses they take time to give the answers. So talk to the course organizers. So you know where the forum is for the course?
No, sir. Actually, sir, I have joined another course and they provide solution after each week. So in this course, I am not getting the solution after each week. So I
Yeah, sure. So you can raise this request in forum. So when you sign into your course, there'll be a section called forum. So under the heading, so under the navigation page, there will be a section called forum. And if you go to the forum, you can post any question you have related to the course, and it will be taken up and answered.
okay, sir.
Yeah. Okay. So let's get back to the question. The cross results in equal number of short and tall plants after a test cross is performed. So we have an answer. So heterozygous parent crossed with recessive parent. Yeah. So essentially, if you are getting all tall parents, sorry, if you're getting all tall parents, then what essentially you're doing is crossing a heterozygous variety with a recessive. So you're crossing with a recessive parent, and that's called a test cross. You really don't know what this genotype is, and you want to see that, right? So essentially, this is what you're doing, and what you will see is uh, if yeah. So essentially, what you'll see is if the unknown variety is heterozygous, you'll see equal number of both the alleles being represented. If it's homozygous, you'll see it go one way or the other way. So if it's homozygous recessive, you'll see all short plants. If it's homozygous tall, you'll see all tall plants. But if it's heterozygous for the height, then you'll see equal number of tall plants to the short plants. So in this case, they're seeing equal number of short and tall plants when they do this test cross. So it's a heterozygous parent. Okay, good. So moving on, the next question, in pea plants, seed shape can be round or wrinkled. and seed color can be yellow or green. Round is dominant over wrinkled and yellow is dominant over green. In a cross between homozygous round and homozygous yellow recessive, which is green actually, so this should be this, or the heterozygous round and homozygous recessive green. How many offsprings will have round and yellow seeds? Can you guys do crossing in Punnett squares and everything? So I will go through how to do this, and you guys can ask me if you guys have any doubt. So essentially, we have Kepler, which is homozygous round, and small y, which is green, and capital R, small r, which is heterozygous round, and small y, which is homozygous green. Right. So these would be the alleles that would be produced. So from the first parent, you get all same type of alleles, which is capital small y, and these would be the alleles that are being produced by the second parent. So half of them would be capital small y, and half of them would be smaller small y. And if you do a crossing, you will see all the offsprings have dominant allele of color, right? So, or round. So, sorry, shape. So, all the offsprings have a dominant allele of the shape, which is capital R. So, there is no wrinkled phenotype that's observed in offsprings when you take this into account. So when you do this crossing, so all of the offsprings will have basically round and round phenotype, right? Now, if you look at the color, all of the offsprings will have the recessive allele for color. So basically, all none of the offsprings will have capital Y because there is no place where they can get a capital Y allele with this parents, right? So both the parents have recessive alleles for the color, so they cannot get the dominant allele for this color. So none of the offsprings will have the dominant allele for this color. So essentially, all the offsprings will be round and green. Now the question is asking how many offsprings will have round and yellow seeds. So none of the offsprings will have round and yellow seeds because the parents do not have yellow seeds. Is that clear? Do you guys have any questions? So you need to have the dominant allele to get yellow seed, and both the parents do not have the dominant allele. So none of the offsprings will have the dominant allele. So none of the offsprings have yellow seed, essentially. Okay. Moving on. A mutant allele where the protein product binds to the wild type protein and inactivates it is a what? Is it an amorph, hypomorph, hypermorph, or antimorph? We've discussed this briefly with the previous question. So in the previous case, there was overproduction of a protein, an active protein. But in this case, what they're saying is there is this protein that binds to the wild type protein and inactivates it. So it's basically uh, preventing the function of the normal protein also. So then what would this be? Is it an amorph, hypomorph, hypermorph, or antimorph? Yeah, antimorph. So uh, it will be an antimorph. So once again, we have different types of mutations. So in case of antimorph, what happens is the mutant protein that's produced inactivates or prevents the function of the wild type. So basically, that's what they're asking in this question. Okay. So I think we're at the end of the previous year's questions. So for those of you that have joined late, yeah, so essentially this week we've talked a little bit about genetics, wherein we've talked about linkage not being explained by Mendel's laws of genetics. In fact, it's one of the drawbacks of Mendel's laws of genetics. We also talked about the mutant varieties of proteins. So in this case, they're asking if a mutant produces more protein, then what would it be? It would be a hypermorph. If it produces no protein, it will be amorph. If it produces less protein, it will be hypomorph. If it produces more protein, it will be hypermorph. If it produces a completely new type of protein, it will be neomorph. And if it produces a protein that prevents or inhibits the action of the wild type protein, then it will be an antimorph. Okay. We've also discussed crossings, several crossings. So in this case, they're asking if two heterozygous parents are crossed with each other, the phenotypic ratio of the offsprings is what? So it'll always be 3:1 if it's heterozygous parents being crossed. And we've talked about independent assortment, and we've also, yeah, so essentially looked at dominant and recessive traits, and we've talked about one the one enzyme that's not a protein, which is ribozyme, and yeah, carbohydrates being the most abundant biomolecule found on Earth. And yeah, again, we've talked a little bit about what a test cross is. So a test cross is any cross that you perform with recessive parent. So it has to be recessive. So in this case, they're doing a test cross for the color. You don't know what this is. So to determine what the genotypic makeup of this is, you will cross it with a recessive, homozygous recessive parent. So small y or green color. And from this, if you get all yellow color, then you know that all of the alleles produced by this unknown plant are dominant in nature. So that would be double dominant for homozygous dominant for color. If it's not dominant, if it's heterozygous, then some of the alleles produced will be recessive. So you will get equal number of yellow and green color. Okay, so that's that, and we've discussed this, I guess, and yeah, that's that. Okay, so do you guys have any other questions that you guys want to discuss? Any topic or questions that you guys might have? Okay, if not, then we can end the session here today. I'll see you guys next week. So take care. No problem. No need to call me sir also. Bye. Okay, see you guys. Bye.